So starting us off today is Professor Theodore Alexandrov, who's been a pioneer in the field of single-cell metabolomics. Last year presenting some really interesting data on activated T cells and how their metabolisms change. And so I'm excited to have start us off today and hear what he's been working on recently.
Cool. Good morning, everyone. Can you hear me all right? Perfect. And I hope also that people who are online, and I was one of those online people last year. So it's a wonderful conference. I hope that you can hear me as well. So my name is Theodore. I go by Theo, and today I'll present to you our work on spatial single-cell metabolomics and how this technology can help reveal metabolic cell states. So, and I spent most of my time in Heidelberg, but recently, last year, we also started commercializing this technology, and we have a second group in Copenhagen at the Bioinnovation Institute, which is also another wonderful place to work. And we hope to push a little bit the needle in this field also from the commercial perspective there as well. So before I start, I would like to present the whole team who has produced all this data, and I'm very happy and proud to lead this team, which combines a lot of people from different backgrounds. And this is what it really needs and takes to develop methods in such field like single-cell metabolomics that you will see is pretty much cutting edge. So we have biologists here, and many PhD students come from biology perspective, and they bring questions. They ask, okay, can we do this? And how we do it, we use mass spectrometry for detection, and here we benefit from tremendous talent and also staff scientists who work and really support all this very complicated instrumentation and advanced, and to really make the best out of it. And then we generate a lot of data, and my background is I come from mathematics and computer science, and this is what often I bring into the projects, really like data interpretation, data analysis aspect. And here we have amazing bioinformaticians and computer scientists who come here, even like from physics, but also computational biology. And we benefit very strongly from devoted software engineers who work pretty much full-time on developing software. And many of them they come not only from biology, but from industrial software engineer perspective. And you will see that this benefits not only us, but also the community. So that's the team. So now about what we do. So over the past years, the focus of our team got crystallized around this phenomenon, metabolic cell state. And by now we know that it is a very elusive phenomenon because of a variety of aspects here, and some of them they are biological, such as it's very dynamic. We know that metabolism is a very, very rapid process. It can happen with a sub-second scale, right? So think about this, how this affects your data. Think about this, how you should plan the experiment in order to be able to really capture that metabolic state which cells had in vivo. And on top of this, definitely, till very recently, it was very hard to detect it because we were lacking the technologies for probing this phenomenon and having the reliable readout for this. We know that metabolic cell state is governed by DNA in cellular programs, of course. But what also interests us and what's unique about the technology such as mass spectrometry is that we can detect also non-genetic factors and their impact on metabolic cell states, such as other cells, maybe neighboring cells, maybe immune cells, maybe we can consider cell-to-cell interactions. Environment, and here we can think about local environment, such as tumor microenvironment. Of course, there is a lot of interest in it, but also global environment. And now with the increase in climate change and climate crisis, we can start this effect as well, such as, for example, temperature. We have a wonderful study with a collaborator where they show that the increase of the temperature just by 2% increases the instability and also triggers the metabolic reprogramming pretty rapidly. So then also disease, right? And of course, reprogrammed metabolism is a hallmark of various diseases such as cancer, Warburg effect, but also we now learn more and more that it's not only cancer which reprogram their metabolism to get, say, to upregulate glycolysis, but also many other cell types. And now the last and maybe one of the most important questions is, how can we develop therapy that would capitalize on our ability to detect this metabolic cell state and our knowledge about metabolic cell states? So this sets the scene for our developments. And how we do it, we develop methods for spatial single-cell metabolomics in order to detect this metabolic cell state in situ, to resolve cell-to-cell heterogeneity, and provide readout which is close to the phenotype for this particular phenomena or for such questions like metabolic diseases. We use mass spectrometry as a detection tool. As you'll see, we develop a lot of software, and now we're getting into this phase and into the time where we actually have access to big data. And here I'm talking about terabytes from thousands of data sets. And now finally, we actually have enough data to start developing machine learning methods or artificial intelligence methods to capitalize on this data, but also to help answer biological questions. Now, moving on, if we start asking questions, why spatial single-cell metabolomics, then pretty much anyone in this field has their own answer. And our response to this question would be that it's two phenomena, and one is tumor microenvironment. And by now, in particular, through single-cell developments and single-cell RNA-seq in particular, we know that it really encompasses multitude of different cell types and cell states and subtypes. But what's even more interesting is spatial organization here, and those non-genetic factors such as pH, right? The cancer cells, they run Warburg effect, they secrete lactate, so it's highly acidic environment compared to a homeostatic state. It's also metabolic waste. It's also lack of oxygen. The solid tumors are highly hypoxic. So how can we actually detect metabolic states of all these cells, how it affects also the treatment? And so this is definitely all the questions where single-cell metabolomics would be very useful. But the second aspect and phenomena that we're interested in, and now maybe even more, is spatial roles of the immune system. And here, about like maybe 5 or 7 years ago, I was absolutely fascinated by now our collaborator Erica Pierce from Johns Hopkins currently. And where in this review with Sue Kayak and others, they discuss immune cells as nomads, which are born in one place, then get educated in another place, and now are attracted by chemoattractants, and they now to come here, squeeze through the blood vessel wall, and completely reprogram their metabolism because they pretty much need to do the same as cancer cells do. They need to highly proliferate here, and they need to exert their function. So they also actually reprogram metabolism, as Erica showed, similar to cancer, to Warburg effect. So, but there is a lot that we don't know, and there is a lot that we can help also, in particular for adoptive cell therapies, to improve their function. So this is kind of like why. Now the question is how we do it. We do it with a technology called MALDI-IMAGING mass spectrometry. And it's very powerful technology, and I'll not go into detail today much, but over the past years was huge transformation and growth of this technology. And now it's really spilling out from academic, from analytical chemistry labs into biology labs. And there is, in particular, because of the vendors who develop better instrumentation here. So we operate technology called atmospheric pressure MALDI 5 Orbitrap. You can see it here. So it is a laser here attached to a mass spec. And if we take a tissue section, we put it in a glass slide, and then afterwards we put it into this chamber, we close the lid. What happens next is the laser will be firing at a particular location. And at this location, there will be also sort of micro explosion. The molecules fly off in the gas phase, they ionized as well. And then afterwards, they will get captured by a mass spectrometer so that for every pixel, we'll have a spectrum. And the spectrum will represent on MS1 level. And the spectrum on MS1 level will represent the relative abundances of our molecules at this particular location. And now we shift a little bit. Then we can shift it 1 micrometer or smaller, but we normally shift 5 micrometer step size. We shift the glass slide, we fire again, we shift, we fire. And this sort of cycle, what we get in the whole data set. And now we can flip this coin or this data set, and we can select not a spectrum, but we can select an MOZ value corresponding to a molecule of interest and construct this image, which would represent the relative abundances of our molecule of interest throughout the whole tissue section. So it is spectral data set, but it's also imaging data set. So, and the properties of the technology that we have is 5 to 10 micrometer pixel size, and this is pretty much the cutting edge. We'll see how it changes like on Sunday, but probably like we'll see definitely 2 micrometer pixel size, if not this year, then probably next year. So then it can detect a lot of molecules, more than 100 metabolites and more than 500 lipids. It can also detect drugs and drug metabolites, which makes it very, very attractive for pharma. Generates a lot of data, and what's very important that of the past decade, this technology, specifically imaging mass spectrometry, specifically MALDI-IMAGING mass spectrometry, became the major technology for spatial metabolomics. So it can generate beautiful images. Just to put an example here, this is a common model, mouse brain. Our lab coordinator, Shiraz, acquired it with 10 micrometer pixel size. You can see anatomical structures. You can see molecules from different molecular classes and pathways overlaid on top of each other. But over the past decade, there was a problem of metabolite identification, very similar to identification problems in other omics. So, but here there are twists why it was specific for this particular technology. And what we have developed here is a solution called Metaspace. And Metaspace by now is a cloud software, but it started as an engine. And for metabolite identification in imaging mass spectrometry, we developed the method for this. And then we implemented it on a cloud using modern software engineering technologies, so that now anyone for free can go there and go to this website, can upload their data set. In 10 minutes, they will get, roughly 10 minutes, they will get their metabolites, they will get images, they can access it through a browser, they can share it, they can organize it, they can do data analysis, and so on and so forth. And this infrastructure started about 7 years ago, and since then it's growing. And in particular, because a lot of users, they use it not only to find molecules, but they use it also to share their data publicly. And this creates this continuously populated knowledge base of spatial metabolomes, in particular because many of these data sets, they come with very useful metadata. And now anyone can go there and mine this data for specific questions which are reflected in their metadata. So just to get you the glimpse how it looks, if you go to metaspace2020.eu, and once again, it's free for everyone. It's open source. So if we select this project or folder of data sets contributed by 2 pharma companies, one Servier from France, another Genentech from the US. So we can pick a particular data set. And these are amazing data sets of whole body mouse sections and images. Now here on the left, we see molecules detected. We can go from molecule to molecule, and immediately here we'll see where this molecule was localized. We can get additional structure information, we can get mass spec information, we can get metadata, and now we also have additional tools for visualization, but also for data analysis, such as enrichment and so on and so forth. And this is just one data set out of more than 8,000 public data sets available there, which were contributed by the users. And this truly put Metaspace into the position of becoming growing global community platform for spatial metabolomics, because we processed a lot of submissions, and many of them are public. It's used by a lot of users. And finally now, in the past maybe 3 years, we started seeing impact of this platform with growing number of publications, which is very, very exciting. So with this, I would like to wrap up the first part of my talk. And metabolism really emerged also over the past decade as a key factor. And there is a lot of interest in metabolism as well in biology, in medicine. So spatial metabolomics has matured tremendously over the past 2 decades. And we have developed Metaspace as big data infrastructure for spatial metabolomics, and soon also for understanding metabolism from this big data. So now moving on, moving away from a little bit spatial, more towards single cell. The question is, okay, MALDI-IMAGING mass spectrometry is cool. Now with Metaspace, we can find molecules, we can go from spectra to molecules, but now how can we go to single cells? And if we look at the history, then single cell metabolomics is actually not new. So if you check the publications in 2011 and 13, there were these amazing reviews, which actually attracted me a lot back then, by Jonathan Swidler and Renato Zenobi, who are probably 2 fathers of single-cell metabolomics. And I was reading this, and I'm like, wow, is it possible to do? I mean, the answer was no. Back then, it was possible to do it only in their labs, right? They were really at the forefront, but it was unreachable for pretty much anyone else. So, and if we look also at the publication, there was really the pause of developments till about 4 years ago. And since then, the technology here really started growing up. And so now, if we also compare it to single-cell proteomics, which is also relevant probably for this crowd to really understand where we stand, is that in single-cell metabolomics, we now stand where single-cell proteomics was about 2019 or 18. So probably when this conference was going through the first or second iteration. So, but it's very, very rapidly growing field, and it attracts a lot of attention. 2 years ago, single-cell metabolomics was recognized among top 10 emerging technologies in chemistry by IUPAC, which probably says something to chemists. So last year was for the first time we have co-organized, and Ryan was, and maybe others, we have co-organized the conference which was focusing on single-cell mass spec, not only proteomics but also metabolomics. And as you will see, it will be growing. And this year, I mean, Nature dropped a big highlight here, recognizing single-cell metabolomics among these 7 technologies to watch in this year. So let's watch it. And by the way, I just want to give a pitch. I think the slide was already previously. Ryan this year is organizing a conference, and we also help, and there is many people they're helping. So and this will also new society. International Society Single-Cell Mass Spectrometry. And the conference will be in Utah. So Ryan is here, so he'll be definitely happy to pitch it and tell more. Okay, so there is quite a lot of attention. So let me show you how we contribute to this field. So we started into this pretty much by chance. It was our first PhD student, Luca, and we were doing metaspace and spatial stuff. And there was already this buzz about single cell going on about 7 years ago. So he approached me. He's like, okay, I'm thinking about my project. Can we do single cell metabolomics with this? I'm like, okay, you can try. So he tried. This completely changed our team, the whole research strategy, pretty much everything. So now almost anyone in our team does projects on single cell mass spec and metabolomics. So Luca developed a method called SPACEM to take the cells, try to grow them on a glass slide or plate them on a glass slide. You can see it also works for non-adherent cells. Then we do microscopy to find where the cells are and to find their also morphometric and fluorescent phenotype and quantify it. Then we run it through MALDI-IMAGING mass spectrometry, run it data through metaspace to get molecular images. Now we have pixelated images, which is not single cell data yet, but close. And then what Luca has developed is a very clever and simple approach how to overlay these 2 types of data with a subcellular precision. And now we can assign the intensities, and through normalization, even multiple intensities to a cell. And now we have truly single cell data, where for every cell here, we have intensities of our metabolites detected, but we also have fluorescent properties, quantified morphometric features and spatial features which cells are next to each other. Now we can start mining this data. And we did it together with Matthias Heichenwälder from German Cancer Research Center to focus on this, what was our first study in single-cell metabolomics. It's about non-alcoholic steatohepatitis, which is a common factor in liver cancer. And it is, if we look at a liver, which upon excess of fat and also glucose, and undergoes lipid accumulation, goes into a fatty liver disease, then triggered by pro-inflammatory cytokines, it actually goes into this inflammatory state called NASH, which is a key factor towards fibrosis and hepatocellular carcinoma afterwards. So once again, we teamed up here with Matthias Heichenwälder and Nira Stadler, who's now actually works in our group in Copenhagen. And they had this beautiful model of human hepatocytes, which are stimulated with the excess of fatty acids and can be also promoted inflammation with a pro-inflammatory cytokine like TNF-alpha. And if we now image these cells and we use lipophilic staining LD540, we see, first of all, that some cells, they're really ballooning with lipid droplets inside, but also we see that even in this isogenic population of cells, there is a tremendous single-cell heterogeneity, that cell next to each other almost doesn't show this phenotype, has almost no lipid droplets. So it was a great model also for us because, of course, if there is a lot of lipid droplets there, the intensities of those lipids will be pretty high in mass spec, right? So, and this is what we did. We considered 4 conditions: control cells, You see almost no lipid droplets here. Then fatty liver disease model with the palmitic acid and oleic acid. Then we stimulated inflammation with a IL-17A. And then here we use TPCA1, which is a small molecule inhibitor of NF-kappa B to suppress the inflammation. So, and we put these cells into space M, and now we can start plotting these single cell plots where one dot is one cell. They're organized here based on the similarity of their metabolic profiles. Here, more than 700 metabolizing cells per lipids per cell. And now we can actually color code them with the conditions. And we see that, yes, of course, expected. The unstimulated hepatocytes, they're different from stimulated ones. But if we now start doing clustering, now we start seeing more fine-grained and interesting details. If we consider this group of cells, which we interpret as residing in steatotic state, based on metabolic profiles, they're mainly occupied by this condition. But we also see a little bit of yellow cells here and a bit of blue cells. Now let's focus on the blue first. The blue ones are treated hepatocytes with a TPCA1, which is anti-inflammatory treatment. Blue ones residing here means that they are non-responders to the treatment, either due to the lack of uptake or due to the lack of response. There is also yellow cells here, which is also very interesting. That means just on the effect of free fatty acids alone, these cells, they reprogram their metabolism to already reside in the state which normally requires pro-inflammatory cytokines. So we see here a beautiful transition which resembles the transition that, so I started with from healthy liver to fatty liver, and then afterwards to NASH. And now we can also start doing this pseudo-time trajectory, we can interpret it in this context. This is a pseudo-time trajectory from least inflamed to most inflamed state. Now we can start showing markers, how they change the intensity, and we can find really markers which are at the beginning of this trajectory or markers which are expressed at the end of this trajectory. And we can interpret these markers, we can find the function enrichment analysis, but what's even more interesting, we can now validate those in vivo model. And this is a mouse model of early NASH, 6 months Western diet. And this here you see bulk lipidomics analysis of liver homogenates. Here you see one dot is one cell, this is our single cell data. And you can see how well the intensities of markers that we find through single cell analysis, how well they were reproduced in the bulk analysis. And moreover, these markers, they were previously reported as predictive markers of NASH in the human cohorts, in human serum. So wrapping up, this shows the potential of using single cell methods to find biologically and maybe even medically relevant markers, even in the human context. So, and this brings me to the summary of the second part. And single cell metabolomics is currently really the emerging field. And so we have developed SPACEM, which is a method for spatial single cell metabolomics. We have shown that it can reveal metabolic cell states in steatosis and NASH. And I'll focus on this later. Okay. So now I would like to take about 5 minutes to present you unpublished results, maybe 10. Present you unpublished results. First, how can we increase the throughput and how can we detect small molecules? Because in the previous part of my talk, I was talking mainly about lipids. And of course, this was mainly lipid reprogramming metabolism, very strong effect. But of course, there is a lot of interest to study other phenomena, which are represented more in the intensity of small molecules. And here I show you the project of our PhD student, Louisa, and lab coordinator, Sharaz, on high throughput single cell metabolomics. So first of all, normally we work with these chambers which are attached to a glass slide, and they work pretty well, but the throughput is very low because it's just 8 samples per slide. So we found an alternative which has 64 microwells, and then we developed the whole system around this, the protocols and software and so on, where we have laser edge glass slides. Now we can put the cells into one of these wells, and we have automated software which processes all these wells. So it's very useful to us because, first of all, it increases the throughput by a factor roughly 5 to 8, and also it reduces the numbers of cells that we need to put there. So now we can work with much more precious primary material. And here we focus exactly on this. We focused on human T cells, and we focused on metabolic reprogramming of human T cells. And if we look what's known about metabolic reprogramming of here, for example, CD4, CD8 T cells, Normally, they reside in this naive state where metabolism is very slow, they're actually not that big as well, but upon activation, so they switch to a highly proliferative and also glycolytic state. So they go to this effector, and then many of these effector cells, they die, and what we have memory cells which go back to reduced glycolysis. So how do normally people study this? I mean, of course, you can study it with proteomics, but many in the lab, when they focus on specifically metabolic effects, they study with this assay called Seahorse. And this is actually a very interesting assay if you don't know it, because it is genius. You have just two readouts. One readout is oxygen consumption, another readout is acidification of your medium. That's it. So how can you study like the state of cells with just two readouts? Because you stimulate them. You give cells glucose and you measure lactate secretion, acidification. If you see that there is increased acidification, that means cells, they really had more capacity to run glucose through glycolysis. Then you inhibit the respiration of the cells, and the cells, in order to get energy, they switch to high glycolysis and they increase even more acidification, right? So it's ingenious readout, and the NSA is adjuvant. So and now pretty much anyone in metabolism lab, they use it. But there is a lot of problems with this. I mean, first of all, it is bulk, of course. It's pretty noisy. It's not that sensitive, in particular for a small population of cells. And of course, it completely lacks the single cell heterogeneity. So, but we took it as an inspiration. And here I'm showing you sort of single cell metabolomics Seahorse assay, where we take a T cell from healthy donors, we isolate them from the blood. This is naive CD4 T cells. Then we stimulate them. Now we have unstimulated, we have stimulated with anti-CD3, anti-CD28, which is pretty much standard immunological stimulation. Now we have stimulate, non-stimulate, and now we use the same inhibitors which are used in Seahorse assay. This inhibitor would suppress the glycolysis. This inhibitor would inhibit respiration. Now we have this full population of cells, and we do single cell metabolomics on those using Spatial. So if we now look at our UMAP plots, one dot is one cell, that we see that naive T cells, they actually have very different metabolisms from activated T cells. And actually, it's a bit surprising because we show that by clustering 97% of cells, they show metabolic reprogramming upon stimulation. So why it's surprising? Because if you ever worked with the T cells, you never see 97%. If you check it by FACS, normally you see 70 to 80%. And I think it brings a very interesting question. Why, if we have all cells metabolic reprogramming, only 70% of those really express the markers on the surface, which are normally used as an indication of the stimulator. And these markers, they are also very functional, right? So there is a gap between what we see in metabolism and the gap what we see in expression of surface markers. And it's not just time, because the cells were stimulated for 3 days. Okay, but now we know that they reprogram, and now we can block the metabolism, and we can inhibit glycolysis. And T cells activated, once again, they actually reside in a very high glycolytic state. That means if we inhibit glycolysis, their metabolic profiles change a lot. So, and we don't see it if we inhibit respiration. They have not that strong response simply because they don't rely on respiration that much. And of course, now we can find markers, and we can do the pathway enrichment analysis and also interpret the metabolic profiles of these cells. But I would like to wrap up with this because I'd like to show you one more snippet of unpublished results. Nikolai, I think I have like 3 more minutes. Yeah. Okay. I think we start a bit later. Good. So then summary of this part is that now we have higher throughput spacer methods, and now very, very exciting. We can detect small molecules. They will be less than lipids. What I've shown was 80 small molecules, but they're very relevant for studying glycolysis, TCA, for a pentose phosphate pathway, amino acid, glutaminolysis, and so on. And what's very, very exciting for us that now we can do this not only for adherent cells, but we can do it also for immune cells in suspension, in particular those such important cells like T cells, like CD4, CD8, or other T cell subtypes. And wrapping up, I wanted to show you the kind of very early results, but I think they may be really relevant for discussions for this conference, is about spatial single-cell multi-omics. And this is a gorgeous project by our PhD student, Bishoy, and actually by another PhD student who is currently here at Broad, Sergio Triana, who kicked off the whole project, then moved here, and Bishoy is now doing an amazing job pushing it to completion. So, and now the question is like, okay, so we did it all in spatial. This was not single cell. We did all in cells. This was single cell, but not tissues. Now the question, can we resolve cell types and metabotypes or cell states in tissues? And here we did consecutive sections approach, where one section we would put the spatial metabolomics, another section we would put the spatial transcriptomics. And here we use Murphy-like approach, which is highly multiplex RNA fish with a subcellular resolution. And here we picked 50 metabolic genes, and we picked also a panel of 50 brain cell type markers. And this was done in collaboration with Resolve Biosciences, which is a company doing this sort of analysis in Germany. So we ran data through Metaspace. We did cell segmentation using bright field, but also using spatial transcriptomic data per se. Then we co-registered this data. And bear in mind that the thickness is 10 micrometer. So co-registration also has some disadvantages. So it's not really that we're hitting the same cell in this section and this section. So we can talk here only about near single cell resolution. But of course, if the region is enriched with a particular cell type, then it probably still has relevance. So now what we have after this co-registration, we have metabolic profile from this cell, and we have transcriptomic profile from this cell, right, which are next to each other in consecutive sections. And we can kind of merge them together. And now we can do correlation between metabolite intensities and cell types and metabolic gene expression intensities. And if we look how it looks, then these correlations, they actually make some sense. For example, one of the highest correlation was between glutamate and this SLC17A7, which has another name as vGlut1. So it's actually glutamate transporter. So we see the link between the function and the metabolites correlated with this. And also what's interesting, if we now start looking at the images between those gene expression patterns and also metabolic patterns, so then we see that spatial metabolomics is not that far away on this anatomical scale, and sometimes has even information, for example, like here, which may be lost in the noisy spatial transcriptomics data. And the final slide that I'll show you is that we were able indeed to link the cell types and metabotypes by first doing clustering of, it's pretty much single cell transcriptomics data just in 100 genes, right? So we did cell types annotation, we mapped them spatially, and we did the same for single cell transcriptomics profile, metabolomics profiles. Now we have this cluster that we call metabotypes, we can map them spatially, and now we can see how they actually relate to each other. And we see that some of them relate. One cell type has actually two metabotypes, which potentially reflects the difference in metabolic states of the cells of this type. And we see that sometimes there is higher resolution on single cell transcriptomic profiles. So sorry, this was pretty quick, but I just want to wrap up, not to take more time. And with a take-home message that spatial single-cell multi-omics, it's absolutely amazing. Once you get this data, it is amazing, combines the best of both worlds, not only for biological and molecular interpretation, but also because you can now link these layers of information about cell types, what we know about cell types, and also the programs, in either transcriptional programs or metabolic programs, or it can be also a protein and protein complexes, of course. So spatial information helps a lot interpret these cell subpopulations. It is completely different world to look at the image or to look at just UMaps. And spatial metabolomics here can help reveal metabotypes hidden from other omics. And with this, I would like to wrap up my talk. I would like to thank Nikolai for the invitation. I would like to thank all of you for the attention. And the whole team who contributed here, because we work highly collaborative. Pretty much everyone contributes to all projects, but there are definitely leaders. And here's Sharaz, Luisa, Andreas, Volker, and Mons, and others, our collaborators on this project. and also past members and the funders. And once again, thank you very much for your attention.
Thanks very much for that really interesting talk. Do we have any questions from the audience?
I have a couple of questions, Theo. The first one is if you can share with us your thoughts about controlling for metabolite changes before they're measured. And the second one is about the prospect of interpreting more spectra than what you're currently interpreting of metabolites for which you probably have good spectra, but we're not yet able to assign them to structures.
Yeah, I think you hit it on the nail. It's one of the most probably important questions nowadays in metabolism, but also metabolomics in particular. So with respect to metabolic changes, yes. So this is pretty much, it's one of the key aspects that we think about it all the time. So we think about this when we design experiments, when we plan experiments, and it's one of the things which actually differs metabolomics and metabolism compared to other omics. I mean, not completely, because like for example, in proteomics, if you start looking at structures, okay, then structures can change also very rapidly and tremendously. So, so what we do, I mean, you need to understand what kind of unwanted changes your metabolites can go through. And this can be, of course, it can be enzymatic, but also non-enzymatic. And enzymatic changes, they are preventable because you can use like agents like PFA, which will cross-link your enzymes and will stop enzymatic degradation. So in this respect, but then you should also understand what happens with your cells and with your tissues when you apply PFA. And also the duration of application is also can be critical. So another aspect is there is a lot of interesting research going on is, for example, like freezing. So freezing, dry freezing, there is a lot of optimization there going on because this is the process which will stop enzymatic degradation, which will stop pretty much metabolism, and will transfer the cells into this like dry state where the metabolites, I mean, they still can undergo modifications, but we'll focus on this later. So that means our take is we think about experimental design, have your controls, think that what can still go with your metabolites and what you are detecting, whether it's reflex or not. That's why it's always important to do validation. I did with some other method, right? About unwanted degradation, it is oxidation. And it is also modifications that we know very little about. For example, now there is a number of studies which show that even at -80, so there is change of metabolites and even lipids in the tissue stored at -80, definitely on different time scale, but we're talking about here more than sample stored for more than a week, right? And this kind of like definitely makes us very anxious, right? So we always try to work with the freshest material possible. And this is kind of concerning the first question. There is definitely more there. And concerning the second question is, yep, we know that so we are running MOSPEC, we're detecting 100 gigabytes of data, I told you, right? And now at the end, I show you 80 molecules, right? So it is definitely very little compared to what's inside. So, and here there are different approaches. So we can improve annotation. And this is what we're doing. We actually at the SMS, we're showing machine learning version of Metaspace. And we show that using machine learning, it is better scoring, it's more sensitive, it like suppresses noise, all these sort of things. We find more molecules. And this will keep going. And the second thing is that we know very little, surprisingly, about ionization, how it happens with molecules. And what are all those peaks like in these gigabytes of data? And this is what often referred as dark matter of metabolomics. And here we also shift a little bit the needle, and others do it much even better by showing that, you know, when molecule metabolite goes in, it's not only protonation, even in positive mode. It can actually get ionized by getting, say, a cluster of matrix, and then protonation, and then lose water, and then have all these very complicated ionization pathways. And now we can actually do this in control scenario, and we show that indeed the metabolites, they undergo these ionization pathways, and by doing this, at least we'll be able to have better quantitation. But there is definitely much more, and that hidden in the data, and here definitely increased sensitivity and increased specificity, like Teams and other technologies. So they will be showing us better detection rates. Yeah.
Thank you.
Thanks for that very thorough, nice explanation.
Thank you. Nice job. Had this one question. You showed us data about difference in FACS analysis versus seahorse, right? When you do with these T cells, and so we look at a lot of immune cells as well in relation to flu. And so I was interested